Well, hello everyone. My name is Mohanish Deshmukh. I'm a professor in cell biology and physiology department. I'm also in the neuroscience center at the University of North Carolina in Chapel Hill. And I am excited to talk about apoptosis today. This is an incredibly fun topic, and we'll discuss three things in today's lectures. First is we'll talk about the definition of apoptosis. I suspect most of you already know this. Second, we'll talk about why it's an important pathway, both in the context of development as well as in several pathological situations. And third, which is the bulk of our lecture, we'll discuss the historical perspective on how the genes were first identified and the actual mechanism by which apoptosis occurs in mammalian cells. Okay, so let's get started. So what is apoptosis? Apoptosis is basically activation of a pre-existing genetic program that results in cell death. So in that sense, it's no different from any other genetic program that the cell can engage, for example, to divide or to differentiate or to move. This program just happens to have the outcome that the cells die. So it's incredibly important for the cells to make that decision very carefully. It's evolutionarily conserved. In fact, people think that all multicellular organisms have apoptotic pathway. And a very important aspect of apoptosis is that the morphological features are very characteristic. For example, the cells shrink, nucleus condenses into small fragments, membranes bleb, and you may have seen pictures of this, and the DNA actually ends up getting fragmented into tiny fragments. And most importantly, I want you guys to remember that it's an active process which requires ATP and in many cases also protein synthesis. And the reason to emphasize this is because, years before, people used to think of death as a passive process, death by neglect, death because cells just starve. Apoptosis is different. This is a decision a cell takes in consideration of all factors, activates the programs, and then executes it where the cells die. Okay, so classically, folks considered cell death in two broad contexts, the context of necrosis and the context of apoptosis. Now, I know that there are many different forms of cell death that have been discovered. You may have heard about them. For today's lecture, we'll discuss on these two broad definitions or two broad contexts of cell death, necrosis versus apoptosis, because I want you all to appreciate the difference. Okay, so what's the difference? So during necrosis, the cells end up swelling and then the membrane ruptures. So this is very important. The cells lyse essentially. Organelles are in disarray. And one very important aspect of necrosis is that there is massive inflammation. Why? Because the cells actually is lyse. And so that's how you end up massive recruitment of immune cells. Typically in an injury situation, when you see redness, that's basically a lot of inflammation because of necrosis. In contrast to this, apoptosis is different, right? So instead of swelling, the cells actually shrink. Organelles are preserved. Cells don't end up rupturing the membrane. The membrane instead blebs. And what I mean by blebs are these structures that are beautiful to look at, and that are engaged because of cytoskeletal changes. Apoptotic bodies, where small bits of the cells actually get separated, are also a feature of apoptosis. Chromatin actually condenses into these small structures, and dying cells can eventually get phagocytosed. Okay, so either it can be professional phagocytes that come and engulf the dying cells, or it can be even neighboring cells. But the important characteristics is that this does not result in massive inflammation, unlike necrosis. I don't want to give you the impression that there is no immune cell involvement. There is, in order to get rid of the dying cells, but by and large, it's immunologically very silent. And that's important because this occurs routinely during normal development. Okay, so when does apoptosis occur? I want to talk about two contexts. First, during development. Okay, during development, this is actually an incredibly important process. From the earliest parts of development, where the developing blastocysts needs to sculpt, there is cell death that occurs during this stage. Another good example is in different developmental stages. There are some cells that are needed at one stage but not needed at another stage. One really good example is the tadpole tail, which is needed during the tadpole stage, but in the adult frog stage, the tail is gone. And essentially, the cells that form the tail, they undergo apoptosis. Apoptosis is also very important in controlling cell numbers. We need to make sure that we have a certain number of cells, not excess, not and so this is controlled by apoptosis. This actually is particularly important in the context of the developing brain. You don't think of cell death occurring in large numbers in the developing brain, but it turns out that that the developing brain, almost half the neurons that you're born with end up dying. And one of the main reasons why almost half the neurons end up dying is because you need to make proper connections, and wiring of the brain is a very complicated task. So you produce more cells, make the perfect connections, and those that are in excess are eliminated. Okay, and so the last one in the context of development is when you're trying to put together a complex organism, some cells are just going to end up in the wrong place. And so you need a mechanism that gets rid of those kind of cells. And so that occurs by apoptosis. I don't want to give you the impression that apoptosis occurs just during development. This is actually an incredibly active process during normal homeostasis. Since the time we started talking about apoptosis a couple of minutes ago, cells in us have undergone apoptosis. And the reason for that is that we are constantly surveying the status of our cells, and we eliminate cells that are damaged, eliminate cells that have become harmful, or eliminate cells that have become infected with viruses or have become cancerous. So this is a very important homeostasis mechanisms that is quietly just trying to fix the problems and getting rid of cells that have the potential to cause harm. Okay, so apoptosis is also implicated in many pathological situations. Okay, I want to give you some examples which occur because of too much and some which occur because of too little apoptosis. Okay, so what are the diseases associated with too much apoptosis? So some examples of that are stroke, spinal cord injury, neurodegenerative diseases such as Alzheimer's and Parkinson's disease. These are complex situations where neurons end up dying. Neurons may end up dying because of multiple different factors, not just apoptosis, but clearly apoptosis is also implicated. Your markers of apoptosis are seen in these situations. And there are efforts to see whether blocking apoptosis can rescue some of the phenotypes of these diseases. It's likely to be more complicated than that. Another good example is AIDS. This is a situation where the HIV virus induces apoptosis of the T helper cells, and that's why you have depletion of the immune system, resulting in the acquired immunodeficiency syndrome. Examples of too little apoptosis is a big one is cancer. Okay, this is a great example because cancers emerge because of a whole different other factors. Cells essentially lose their ability to control their cell division. There's excess cell division, but a common feature of all cancers is that the cells should have undergone apoptosis, and they don't. Okay, so the ability to eliminate any abnormal cells is a key feature of apoptosis. When that doesn't occur, then you can have situations such as cancers emerge. And in many of the exciting medicines that are being developed for cancer therapy, particularly what people refer to as personalized therapy, they are trying to figure out exactly why the apoptotic pathway is not able to be activated and then force its activation. Other context is autoimmune disease. This is a situation where the normal development, the self-reactive cells are eliminated, and that elimination occurs by apoptosis. If the apoptotic pathway is blocked, then that can result in autoimmunity. Okay, so here is a historical timeline of apoptosis research. I've taken the liberty to make this timeline. I've broken it into 4 different sections because I want you guys to get an appreciation for how this field had developed. Now, the first thing is literally hundreds of years ago, scientists had been studying and defining a form of cell death that had common morphological features during development. I'm sure some reports might even go before 1800s. And when electron microscopy was developed, they had used that as a tool to define this particular morphological characteristics of cell death that occurs during development. What happened in 1972 was a landmark paper was published by Kerr, Wiley, and Curie, and that's the paper in which they defined the term apoptosis to this type of cell death. Okay, so this is why I put a historical context of 1972. From here on, the word apoptosis became popular in the context of defining this type of cell death. So then for about 20-30 years, one of the most interesting things that happened is that the genetic pathway of cell death was discovered, as we'll discuss in the next slide, first in the context of C. elegans. BCL2 caspases were identified as regulators of apoptosis in mammalian cells, and also the death receptor pathway and the Fas ligands and the TNF receptor were also all defined to be important features of apoptosis. I put an emphasis on 1996 because another landmark paper was published in 1996. And what this was done by Xiaodong Wang's lab, was that his group identified cytochrome C as a key activator of apoptosis. And this really made mitochondria as a central player in apoptosis. And from here on, mitochondria became recognized as a key regulator of apoptosis. We appreciated the increasing complexity of apoptosis, expanded the function of BCL2 family proteins, particularly for BH3 family proteins, IAPs, and of course, the exciting research on this continues to this day and in the future. So the mutagenesis screen in C. elegans led to the identification of 3 genes that regulate cell death in C. elegans. Okay, these are called the SED genes, SED standing for cell death abnormal. And 2 of these SED genes, SED4 and SED3, were required for cell death. In other words, if you had mutant worms that were mutated in either SED4 or SED3, then these worms, the cells in these worms, did not die. In contrast, SED9 is an inhibitor of cell death, and so mutations in SED9 caused more cells than normal to die. And so this, of course, a few other genes have also been identified since then in C. elegans, but this is the core pathway of apoptosis in C. elegans that was identified through the mutagenesis screen. Now, what's striking about it is that there are homologs of all 3 of these genes that regulate the cell death pathway in mammalian cells. And at the main fundamental level, the structure kind of is the same. So what I mean by that is the homolog of SED4 is APAF1, and a homolog of SED3 are caspases. And so both APAF and caspases are required for apoptosis in mammalian cells. And the homolog of SED9 is the BCL2 family genes. And the BCL2 family genes, at least the initial member, BCL2, is an inhibitor of cell death. Things in mammalian cells have evolved into having multiple members, and we'll talk a little bit more detail about that. The BCL2 family now has both pro and anti-apoptotic members. Caspases are all, it's a large family, which is all inducers of apoptosis, and APAF, to the best of our knowledge, is still a single gene family. But I hope you guys appreciate how the initial identification of genes in C. elegans really paved the way for the identification of the genes regulating apoptosis in mammalian cells. So for the rest of the lecture, we're going to focus more on the pathway of apoptosis in mammalian cells. Okay, and typically one would start with the pathway from outside to all the way inside, and what happens in the end. But in this context, I actually want to start with inside out. So let's look at what are the key features of apoptosis, and then let's make our way backwards to see how are these key features of apoptosis brought about. So what are the key features of apoptosis? As we learned earlier, the dying cells exhibit fragmentation of DNA, chromatin condensation, membrane blebbing, so this involves cytoskeletal changes, and in the end, phagocytosis. And the family of genes that effect these changes are actually the caspases. We talked about SED3 and mammalian caspases. Well, this is where the caspases come in. The caspases are among the last is among the last event to happen during apoptosis. And once caspases are activated, at least the ones that are involved in executing cell death, once those caspases are activated, then they can actually effect these apoptotic changes. Okay, so what are caspases? Well, the caspase family is a pretty large family in mammalian cells. There are at least 12 known caspases in mammalian cells. Some of them are involved in apoptosis, some are involved in inflammation, and others could have some other functions. So just to keep it simple, we'll focus on broadly caspase family, but then talk mostly about the ones that are known to be involved in apoptosis. So caspase family structurally has two subunits, so the P20, so the large subunit and a small subunit. And most of caspases have a prodomain. Now this prodomain can be relatively short or can be much longer. And it turns out that the caspases that have the longer prodomain, actually the prodomain is very important in activation of caspases. Okay, so caspases, as I said, are the mammalian homologs of the C. elegans SED3 gene. They're basically cysteine proteases. What that means is that their active site is a catalytic cysteine, but their specificity is that they cleave after aspartic acid residues. Okay, so not every aspartic acid residue in their substrates, but they utilize a substrate sequence specificity in which proteins are going to be cleaved. For example, a executioner caspase called caspase 3 cleaves after the sequence DEVD. Okay, whereas another caspase called caspase 1, which is involved mainly in inflammation, cleaves after the sequence YVAD. But the cleavage is after aspartic acid residues. And what these caspases do is that once they're activated, they can cleave specific cellular proteins to induce rapid apoptosis, at least the ones that are involved in apoptosis. And so if you ask me how many proteins are actually cleaved by caspases during apoptosis, that number may well be in the hundreds, if not 1,000. So a lot of proteins. There are some key ones that we'll talk about, but a lot of cellular proteins seem to be cleaved during apoptosis. And the consequence is that the cell really ends up getting dismantled and dies quickly. Okay. So the caspase family that mediate apoptosis can be roughly divided into two parts. So they are the initiator caspases. As the name suggests, these are the caspases that initiate apoptosis. And there are the executioner caspases. And these are the caspases that actually do the job of killing the cell. In the context of initial caspases, the two that have received the most attention are actually these two, caspase 9 and caspase 8. And I should mention that one of the distinguishing characteristics of the initiator caspases is this presence of a larger prodomain. Okay, so this is the sequence that's in front of the large domain. You can see these initiator caspases contain one of two kinds of domains, either the card domain, which is called the caspase activation and recruitment domain, or the DED domain, and that's referred to as the death effector domains. And what these initial caspases do is utilizes these domains, either the card domain or the DED domain, to basically interact with other protein that form a complex in which these caspases are activated. So caspase 9, for example, is activated on the apoptosome complex via, as you will see, interaction with the card domain, and caspase 8 is activated on the death receptor domain via the DED domains. These interactions lead to the activation of these initiator caspases, and once these initiator caspases are activated, they can cleave and activate the executioner caspase. The one that has received the most attention is caspase 3, but there are other contexts in which caspase 6 and caspase 7 are also activated. But once you have the executioner caspases activated, and as I said, these initiator caspases cleave and activate the executioner caspase, once you have caspase 3 activated, for example, then it cleaves hundreds of cellular proteins to induce rapid cell death. I want to talk about two examples just to show you how the activated caspase-3 can exert these apoptotic changes. Okay, so the first example focuses on how you get DNA fragmentation. And the enzyme that causes DNA fragmentation is an enzyme called caspase activated DNA. It's called CAD. And this enzyme is normally in a complex with its inhibitor called ICAD, so inhibitor of caspase activated DNAs. And this is the normal situation in a healthy cell, because it's very important for this enzyme to be in a complex with its inhibitor, because otherwise you would get DNA fragmented all the time. And so what happens is that once you have caspase-3 activated, this can actually cleave the ICAD, and you essentially have the release and activation of, not activation, just release and activity of CAD, which then cause DNA fragmentation. So this is how caspase-3 can actually cause DNA fragmentation. In another example, caspase-3 can actually cleave another protein, for example, called ROC1, so Rho activated serine threonine kinase. This protein is normally in an inactive conformation, but cleavage of caspase-3 causes it to become active. And then once ROC1 is active, it can phosphorylate the myosin regulatory light chain that then causes membrane blebbing. So here's a situation where the cytoskeletal changes are mediated by caspase-3 via activation of the ROC1 kinase. And so these are two just examples of what happens when you have caspase-3 activated. Okay, so let's talk about how these caspases, caspase-3 in particular, is activated in the context of apoptosis. We know what it does once it's activated, but let's talk about how caspase-3 is activated. And there are two main pathways. First, we'll talk about the extrinsic pathway. Okay, it's also called the death receptor pathway. The reason it's called the death receptor pathway is because there are molecules like FAS, for example, or TNF, tumor necrosis factor. Both these can actually bind to their receptors. FAS will bind to FAS receptor, TNF will bind to its TNF receptor. And once these ligands bind to its receptor, what happens is that these receptors get trimerized, and then FAD gets recruited to these death receptors. Okay, FAD is a adapter proteins, and what this adapter protein does is it helps recruit caspase-8 to this death receptor complex. And this interaction occurs via these DED domains that we talked about before. Remember, caspase-8 is a initiator caspase. It has these DED domains. With the DED interaction, helps recruit caspase-8 to the death receptor. And once caspase-8 is recruited to the death receptor, it gets activated. Okay, it gets activated by a process called induced proximity. And once caspase-8 is activated, what it does is that it can directly cleave and activate caspase-3. And so this is actually the fastest way to initiate apoptosis in a cell. And because it's a direct short circuit from the external receptors directly to the caspase-3. And so this is called the death receptor pathway, or the extrinsic apoptotic pathway. Okay, it's the fastest way to kill a cell. It's usually employed by immune cells when they need to do something fast. If they're infected with a virus, for example, they need to execute the death process very quickly. Another way of activating caspase-3 is via the intrinsic pathway. This is also called the mitochondrial pathway of inducing apoptosis. And in that context, the initiator caspase is caspase-9. Okay, so not the death receptor, but caspase-9. And so there's an incredibly interesting mechanism by which caspase-9 is activated in the intrinsic pathway. And this really came as a big surprise in the field, because it turns out that the key trigger for activating caspase-9 was this protein in the mitochondria that is called cytochrome C. Cytochrome C had been well studied in the context of mitochondrial oxidative phosphorylation. It's actually a key protein for generating ATP and for survival. And it turns out the very same protein that helps you survive and generate ATP actually in the context of apoptosis comes out. Okay, it comes out, it's triggered out and then that cytochrome C, what happens is that it binds to a protein called APAF1, and then there's this complex called the apoptosis complex that's formed that results in the activation of caspase 3. Remember from the C. elegans slide, I'd mentioned that the mammalian proteins APAF and caspases are important. Well, this is how it fits in the context of the mitochondrial pathway. And this really was a major surprise because nobody considers cytochrome C to be a key trigger of cell death. And it's one of the most fascinating aspects of the cell death, mammalian cell death field. Again, the mechanism by which caspase 9 is activated on the apoptotic complex is via interaction with the card domain. So it turns out that APAF also contains a card domain, caspase 9 contains a card domain, and these card-card interaction, again, on this complex causes the activation of caspase 9 by a process that people refer to as induced proximity. But once caspase 9 is activated, then 3 is activated and the cells die. So what regulates whether the cytochrome C is going to stay in the mitochondria or comes out? And that turns out to be the function of the BCL2 family of proteins. The original member of the BCL2 family was called BCL2, B-cell lymphoma. But now this family has evolved into multi-members. We'll talk more about it. But the important thing to keep in mind right now is that this is the family that is the gatekeeper of determining whether cytochrome C stays inside or comes out. And there are two key proteins in this family that basically help cytochrome C come out. But in a normal, healthy situation, BCL2 makes sure that cytochrome C stays inside the mitochondria. And if you're wondering what are the external or what are the stimuli that help BCL2 determine whether it should promote the release of cytochrome C or not, these are the stimuli that are the triggers, various triggers for apoptosis. For example, growth factor withdrawal in neurons, for example, or DNA damage in virtually any cell, stress, ER stress, for example, or other stimuli. Whenever cells are faced with these kind of stress stimuli, they basically figure out whether they can cope with it, whether they can fix it, or whether they should induce apoptosis and die. And if they decide to induce apoptosis and die, then these signaling pathways, they use different components to signal to the BCL2 family. We'll talk more about it very soon. But once the BCL2 family proteins are sort of urged to induce this release of cytochrome C, then it's the common pathway of apoptosis in all these different situations. And as I said, that's called the intrinsic pathway of apoptosis. So basically, just to summarize, you have two ways of activating caspases in cells. The fastest way is through the death receptor pathway via caspase 8 as the initial complex. This is also called the extrinsic pathway. Or there's the mitochondrial pathway, the intrinsic pathway. The key trigger here is cytochrome C and the apoptosis formation. But the outcome then is still activation of caspase 3. So this is basically the entire picture in a nutshell. It looks complicated, but I hope you guys appreciated how all these pathways lead to activation of caspase 3. That kills the cell, and then there are two ways of reaching this point. Okay, so in the next slides, we're going to talk about a little more detail about how the BCL2 family is regulated and other ways of regulating caspases.